Good morning, students. My name is Prof. Teane from the Department of Adult Community and Continuing Education, UNISA. This morning, I just want to share with you, the grade 12 learners, one of the topics in life sciences called DNA, the code of life. This is what we are going to talk about this morning regarding the topic. We are going to look at nucleic acids because DNA forms part of this. Looking into the structure in details, the DNA profiling, the structure of RNA, DNA replication, and protein synthesis. What are nucleic acids? Nucleic acids are biomolecules made up of nucleotides that stores and transfers genetic information. We have two types of nucleic acids, that is deoxyribonucleic acid, abbreviated as DNA, and ribonucleic acid, abbreviated as RNA. And let me just show you how different the two nucleic acids are. Look on your left-hand side, we have two diagrams that showed RNA and DNA. Automatically, you can see that one of the nucleic acids has got one strand and another one two strands. And the one with one strand is RNA, and the one with double strands or a double helix is DNA molecule. So RNA is very short as compared to DNA, meaning DNA is a little bit longer, and RNA is made up of a sugar called ribose sugar, and DNA sugar is deoxyribose sugar. As we continue with our lesson, you are going to see more of the differences between the two nucleic acids. For now, let's look at the next slide, which shows a nucleotide. Now, what is a nucleotide? Nucleotides are building blocks of nucleic acids, meaning the DNA and RNA molecules are built up by nucleotides. Each nucleotide has got three most important parts, which is the phosphate ion, a sugar called pentose sugar, and the nitrogenous base. Let's look here again. Remember, I mentioned that nucleotides are the building units of nucleic acid. And one of the parts of a nucleotide is the nitrogenous base. So on this slide here, I'm showing you the nitrogenous bases, both for DNA and RNA. Now we have two types of nitrogenous bases. They are the purines and the pyrimidines. The purines are adenine and guanine, whereas pyrimidines are cytosine and thymine. Right from the beginning, I just want you to locate this, that in both RNA and DNA, the purines are the same, meaning both of them have adenine and guanine. But When coming to pyrimidines, the pyrimidines for DNA are cytosine and thymine, whereas in RNA we have cytosine and uracil. So we don't have thymine in RNA, instead we have uracil. Same applies to DNA. We don't have uracil, we have thymine. Now let's come to the business of the day. Remember, I said the topic is about DNA, the code of life. Let me remind you a bit on where this DNA is located within a cell, right? I'm taking you back to your grade 10 syllabus, where you were taught the structure of a cell, both plant and animal cell. Now for this topic, let's look at the animal cell. We know that one of the parts of an animal cell is the nucleus, which we know it is the part that carries the hereditary characteristics of a cell. And inside the nucleus, we have chromatin network, which will change during cell division into chromosomes. So chromosomes emerge from this chromatin material. And it is these chromosomes where we find the DNA molecule. And a section of a DNA molecule is a gene. So I repeat again, DNA is found in a cell where inside the nucleus, where specifically in the chromosomes, and the DNA is made up of genes. In the nucleus of almost every cell in your body is the collection of DNA needed to make you as an individual. So it is found in the nucleus of a cell as already indicated. Now, the chromosomes that I talked about, which emerge from the chromatin for a human being, every normal human being has 23 sets of chromosomes. All in all, there are 46. But always, these chromosomes are found in pairs. Why? Because we have a donation of 23 from our mother, another 23 from our father. So they are found in pairs as indicated in this slide on your right hand side. Chromosome has got the DNA, which I have already indicated is made up of genes. And the number of genes, the total number of genes, what we call the human genome, is estimated to contain roughly 20,000 to 25,000 protein coding genes, which are segments of a DNA molecule. Remember what I said when we started, as I was showing you the structures of the two nucleic acid. I indicated to you that RNA has got single strand and DNA has got double strands. So this is the structure of a DNA molecule. Now the purines and the pyrimidines that I talked about when I started this lesson, indicating to you that the purines for DNA are guanine and cytosine, and the pyrimidines, thymine and adenine. Now, in the structure of a DNA molecule, it indicated to you that the DNA molecule is formed by the nitrogenous bases, which are purines and guanine. So the guanine in the structure of a DNA molecule will always match with cytosine. We normally use the letters G for guanine and C for cytosine. And thymine, which we give the letter T, will match with adenine. So if you look at these nitrogenous bases, always the purine will match with a pyrimidine. Now, how possible that always guanine will match with cytosine and thymine with adenine? It is because of the structure. If you look at this diagram here, you will realize that, look at this red picture that could represent any of the nitrogenous bases. But for argument's sake, let me say the red part here represents guanine and the yellow part cytosine. So the shape of guanine is similar to the shape of cytosine, cytosine. So they both form what we call a lock and key order. So there will be no way where we find that guanine is matched with, for example, adenine or cytosine because of the shape. It is shaped in more or less the similar way to fit in either the purine or the pyrimidine. And the two are being joined together by a bond called hydrogen bond. And let me mention this right from the beginning, that this is a weak bond. When I say it's a weak bond, I mean that it can easily break. It can easily break. Now let's look at the blue one, and let's give the blue structure the name thymine or T, and the green one for adenine. Look at the shape. It fits in to the complementary nitrogenous base. So there will be no mistake. And this is the bond, which I said it is a weak bond. All right. So this nitrogenous base and this part here, which is the sugar, and remember what I said about a nucleotide? And, all right, nitrogenous base, meaning the purine and pyrimidine will match. The purine will match with a pyrimidine. If this red part is guanine, then the yellow part will be cytosine, and they will be held together by a weak hydrogen bond. Same applies this side. If this is thymine, the blue part is thymine, the green part will be adenine. So the reason why there will be no mistake, always guanine will match with cytosine and thymine with adenine, it is because the shapes of these nitrogenous bases, the purines and the pyrimidines, fit in, meaning a purine shape will be in such a way that it will form a lock and key order with a pyrimidine, a complementary pyrimidine. Guanine will always match with cytosine, and thymine will always match with adenine. Okay, I said to you that a DNA molecule and an RNA molecule are formed by nucleotides. The building unit is the nucleotide. So the structure here, this red part shows a nitrogenous base, and this part is a sugar, and this is a phosphate. So a phosphate, sugar, and a nitrogenous base form what we call the nucleotide. As I said, DNA is formed by nucleotides. Now, we have already indicated the sugar that forms a DNA molecule. It is called a deoxyribose sugar. So this sugar here is a deoxyribose sugar because this is the structure of a DNA molecule. So when you look at this diagram, you will see that there's a repetition of a nitrogenous base, a sugar, and a phosphate. Quite a number of nucleotides form a DNA molecule. So this is a phosphate, as I've already said, and this is a deoxyribose sugar. All right. Then we have the name here of the nitrogenous base. T stands for thymine. So the red part here shows the nitrogenous base, thymine. And as already said, thymine will always match with adenine. And guanine will match with cytosine. So remember, in the structure of a DNA, thymine matches with adenine, whereas guanine matches with cytosine. Just look at this little picture, reminding you of how the purines and the pyrimidines are matched. Thymine goes with adenine and guanine with cytosine. Okay, I'm very sure that when we look or we watched our TVs, we are always being confronted with shocking news about crime in our country. And you have heard that, I think, on several occasions, when people are being killed, are being raped, they do what we call DNA profiling. And what is DNA profiling? This is a specific type of DNA test used to identify individuals based on unique patterns in their DNA, especially in non-coding regions, such as short tandem repeat or STRs. So it is used in forensics, crime scene analysis, can be used for paternity and maternity, and identification of the victim's DNA when victims are exposed to disaster, like floods, like fire. Now let's look at these STRs. If, for an example, there's a woman who has been raped, and then they collect semen, and there is 1, 2, 3 suspects brought forward to say one of them or all of them might have raped this woman. What they do is they collect semen from the woman and check this short tandem repeat. They look at this DNA pattern. So this is what they shall have collected from the semen, and then they will then make DNA tests for all the 3 suspects. Look at these pictures very well. You will see that the DNA from the semen, that is the crime scene, is similar to suspect number 2, which means that suspect number 2 is the one that committed this crime. So DNA profiling is very much important. And sometimes we find people, especially the young boys, saying they are not the one that pregnanted a woman. And the DNA profiling can also help to determine whether the boy or the man is the father to this child. Same applies to young girls who would just leave their children in the bush, in the toilet, everywhere. They can take, if there is a suspect, they're able to take the DNA test of the mother and check as to whether it matches with the child. So DNA profiling is very much important. So the study of DNA is very much important. We don't study DNA just to pass our matric. It is useful in life, and you can even help people out there who do not know about DNA and DNA profiling. Another nucleic acid is RNA. Look at the nitrogenous bases. U stands for uracil, and G for guanine, A for adenine, and C for cytosine. Remember I said guanine, adenine, and cytosine are also used to form the structure of DNA. But instead of uracil, DNA has got thymine. So RNA does not have thymine. Instead, it has uracil, but also have what DNA has, which is guanine, adenine, and cytosine. There's the structure of a nucleotide here. This is a nitrogenous base. This is the sugar, and this is the phosphate. And remember, the sugar for RNA is the ribose sugar. Remember, the full name for RNA is the ribonucleic acid. So RNA is a single strand. It has got the sugar called ribose. It has got 4 nitrogenous bases, adenine, which will always match with uracil, guanine, which will match with cytosine. RNA has got, we have 3 types of RNA. the messenger RNA, which is written as M, small letter, RNA, transfer RNA, small letter T, then RNA, ribosomal RNA, small letter R. The messenger RNA, you will learn more about messenger RNA as we tackle the section on protein synthesis. You will know more about the messenger RNA. For now, let's skip this. And also the transfer RNA is another form of RNA. For now, let's skip this. We'll come back to the role of each of these RNAs. This is the ribosomal RNA. Okay, let's quickly look at the two nucleic acids, the differences between DNA and RNA. DNA is a double strand, RNA is single strand. DNA is a little bit longer than RNA. The sugar is deoxyribose sugar. RNA is a ribose sugar. We have these nitrogenous bases in DNA, thymine, which will always match with adenine, guanine, which will match with cytosine, and RNA. Guanine will always match with cytosine. Thymine will match with uracil. So in the structure of DNA, the bases are paired, whereas in RNA, there's no pairing of bases. Okay. DNA has got one specific characteristic which we don't find in RNA. It can replicate. It can duplicate. What is the meaning of this? This is the process whereby the cell makes an exact copy of its DNA during the cell division, both mitosis and meiosis, in the interphase stage. DNA can form another DNA which is identical to the original DNA. And what is the reason for this? This is to ensure that each cell that is being produced contains the same DNA. So during mitosis and meiosis, cell division, there's a stage called interphase, the very first stage. And that is the stage where DNA replication takes place. DNA duplicates itself or divides into two identical DNA molecules, making the exact copy of itself. Now, how does that happen? During the interphase, the first stage of meiosis, a DNA molecule will unzip. What does that mean? Remember, I've indicated to you that the purines and pyrimidines are joined together by a weak hydrogen bond. So when DNA is ready to form a replica of its own kind, the hydrogen bond will break one by one, separating the purines from the pyrimidines. We call this process the unzipping, because this breaking down of the hydrogen molecule does not happen at once. It happens like a zip of a garment. when you unzip whatever garment. So when the hydrogen bonds of the entire DNA molecule breaks, we will have 2 strands of DNA. Remember this process, let me keep on reminding you that this process takes place inside the nucleus, where we find the DNA molecule. Guanine will be separated from cytosine, thymine from adenine, and at the ultimate end, we're going to have 2 strands of DNA molecule. It's no longer a double helix, but there are 2 strands that are not joined. Then what happens? Inside what we call the nucleoplasm or nucleocell, which is the solution or liquid found inside the nucleus, there are loose nucleotides. Remember, a nucleotide is used to form a DNA molecule. So the nucleotides will arrange themselves along the 2 strands. There will be no mistakes. Remember what I said? The shapes of the purines are similar to that of the pyrimidines. They sort of form a lock and key order. They're designed in such a way that they can form a lock and key order. So in both strands, nucleotides will align themselves according to the template maintained by each strand. And finally, we will have 2 DNA molecules that are identical, that are even identical to the original cell. But sometimes there could be some mistakes or changes. We call that mutations. We'll talk more about mutations when we deal with topics like genetics. For now, you must know that it can happen that, instead of a nucleotide, which is supposed to be arranged in the same way as the template DNA strand, something can happen which is very strange. Nobody controls it, and a mutation can happen, meaning the structure of this DNA shall have changed. It is not the original DNA. We'll talk more about that.